Hello friends, welcome back to my YouTube channel, The Microbiology Digest. So we have discussed already the initial chapters of immunology. So today we will be discussing a new topic, that is the immunity and its types. What is immunity and what are the different types of immunity? So what is immunity? We have already discussed. It is ability of our body to protect ourselves from the infections. But there are different phenomenons are involved and different processes are there. So we'll be discussing one by one. Now today let us understand what are the different types of immunity. So immunity, as we have already discussed, that this is the ability of our body to protect ourselves. So there are mainly two types of major types of immunity are there. First one is called as innate immunity. This is also otherwise called as natural or non-specific immunity. And the second type, it is called as acquired immunity or adaptive immunity. So immunity, the we can divide into, as I said, two types: innate immunity and adaptive immunity. This adaptive immunity, further, it has been subdivided into two types. It is natural or artificial. Again, artificial as well as natural may be of active type or passive type. Again, the artificial may also be active or passive type. Now, let us understand what is innate immunity. Innate immunity, it refers to the non-specific defense mechanisms that come into play immediately. We can say within hours of exposure to an antigen. This innate immunity, this may be conferred by different types of barriers actually. So these barriers, there may be four different types of barriers: anatomical or physical barriers, then physiological barriers, phagocytic or cellular barriers, and the inflammatory cells. So these barriers, first they will try to eliminate the infectious agents or antigens from our body. So innate immunity, as I said, this is non-specific. So it has no relation to the previous exposure to any antigen. Okay. So immediately after exposure to an antigen, our body responds through the innate immune mechanism. So this is non-specific and it is conferred by different types of barriers, mainly anatomical barriers, physiological barriers, phagocytic cells. They will try to eliminate the pathogen, and finally the inflammatory cells. This innate immunity, it acts as a first line of defense immediately, as I said, immediately after exposure or entering of an antigen to a body. This innate immunity, it acts as a first line of defense to eliminate the pathogen from the body. Now, what are the properties of innate immune response? So, innate immune response, as already we have discussed, this is independent of antigen exposure. Means innate immune response, it does not require any previous exposure to antigen. Then it is immediate response. It is the body responds to immediately to an antigen. Maybe within hours, this response come into place. Then this is non-specific. This is, as I said, this is also otherwise called as non-specific immunity. Then last one, very important, that is the innate immunity has no memory. Means no memory response is generated in the non-specific immune response. So in the table you can see the summary we have what are the different mechanisms by which innate immune response is generated. So there are, as I said, anatomical barriers are there, physiological barriers are there, then phagocytic barriers are there, then inflammatory barriers. These barriers collectively they will come into play and they will try to eliminate the pathogen. Now, how these invaders are recognized by the body means when our body identifies how our body identifies a pathogen. So these invaders means these are the pathogens or microorganisms which comes inside our body and they can establish an infection. So body has to identify these organisms or these invaders. Only after identification or identifying these uh, microorganisms or these invaders, the body will respond to it and it will try to eliminate. Now, this recognition step, this identification step is the first step in the innate immunity. First, when innate immune response is mounted, so body has to identify the object or the invader. So this identification or the recognition is through it is done through some of the cells which are called as sentinel cells. Sentinel cells, for example, macrophages, dendritic cells, mast cells. So these cells, different functions of these cells already we have discussed in our previous discussions. So these cells actually they use receptors. They can recognize highly conserved molecules or molecular patterns. Collectively, they are called as pathogen associated molecular patterns, or in short, they are called as PAMPs. So these cells they use some of the receptors. Those receptors they can recognize these patterns, PAMPs which are present in the pathogens or microbes. So these PAMPs are present in the microbes, and these PAMPs, pathogen associated molecular patterns, can be recognized by these sentinel cells through its receptors. So they are PAMPs are common, essential, and conserved molecules or structures which are present in the microbes, or it is produced by a diverse range of microbial invaders. So these PAMPs they can be recognized by our sentinel cells, and these PAMPs, for example, PAMPs include the lipopolysaccharides which are present in the bacteria, peptidoglycans same also present in the bacteria, and nucleic acids which are present both in bacteria as well as other microbial invaders. Then if suppose these invaders are not microbes, they are some of the cells which are uh, present released from our damaged tissue. So these they can also recognize these patterns, molecular patterns. They are called as damage associated molecular patterns. In short, they are called as dumps. Okay, so both these pumps and dumps they have their receptors. They get attached or they are recognized by the receptors which are present in the sentinel cells. These receptors are called as pattern recognition receptors, or in short they are called as PRRs. PRRs, they are present in the sentinel cells, and through these PRRs pumps and dumps can be recognized, or they can identify, and in turn our body can identify the potential invaders which are coming to our body. Next, mm, this what is this pattern recognition receptors? One of the very important example of recognition receptors are toll-like receptors, or in short they are called as TLRs. So these are the most significant family of pattern recognition receptors. Their name actually it is given from their original discovery of a protein called toll in fruit flies, and these are found, TLRs are found in the sentinel cells of the innate immune system, and these TLRs, they can bind to pumps, and they can give the signal to our body that this is a invader, foreign invader, and action should be initiated against it. So there are different types of TLRs have been identified so far. It is around 10 to 12 different types of TLRs have been identified, and these TLRs, they are responsible for binding with the pumps and thereby giving the recognition to the for identification of the potential invaders or antigens. Now this is the table showing the TLRs identified so far, their location, and the ligand to which they binds. Most of the TLRs, they are present in many of the microbial organisms. TLR 1, 2, 3, 4, 5 up to 13 so far they have been identified, and they have specific ligands to which they binds, and they help in initiation or mounting the innate immune response. Now this is how the innate immune system works, the flow chart. First, the pumps and dumps will be recognized by the pattern recognition receptors. They will be binding, then they will produce a signal. This signal will in turn cause the release of cytokines or other antimicrobial molecules by the body, and it will initiate for the destruction of the invader. Next, another very important phenomenon of innate immune system is the inflammation. So inflammation, it is actually it is innate immune system, as we have already discussed. It consists of diverse subsystems which work one by one for elimination of the invaders. So it is inflammation, it is a process whereby defensive cells and antimicrobial molecules converge on sites of microbial invasion or tissue damage. Okay, so inflammation, it is the process where various cells or cells of the immune system, defensive cells we say, then antimicrobial molecules, they come into the site for destruction of the invaders. So this inflammation, it is a sequential step process, one by one, after first recognition of the invaders through pumps, and then it will cause vasodilation and release of the cells. So this is a complex process. Finally, it will cause destruction of the pathogen or invading microbes. Now there are specific signs which we call as cardinal signs. Once inflammation process is initiated, it will show four different types of cardinal signs. One is called as rubor. It is the redness, means the site at which inflammation is triggered, it will become red. Then next is the calor, means temperature. So the site where inflammation is triggered, it will be hot. Then the dolor, it will be painful. That site will be painful. And last is the tumor, means there will be swelling of the area. So if these cardinal signs are seen, we can say that inflammation process is already initiated, and it is a part of an important part of innate immune system for elimination of the invading pathogens. Another major component of innate immunity is the phagocytosis. Phagocytosis means once sentinel cells recognize the invader, they will generate signals and activate the other cells which will act on the invader for elimination by killing. So phagocytosis, actually this is the process by which the cells, they can kill the pathogen and they can cause destruction of the same pathogen, and in turn it will be eliminated from the body. Phagocytosis, actually the term phagocytosis, it is coming from the Greek word which means eating by cells. Means this is entirely a cellular process where the cells, they will initiate the killing process of the pathogen and the destruction of the pathogen. Then the process of phagocytosis, it has different discrete stages, namely activation, chemotaxis, adherence, ingestion, and finally the destruction. So all these processes, they sequentially one by one it will come into place, and finally the invader will be killed and it will be eliminated. So this is how actually phagocytosis occurs. Major action is taken by the neutrophils. That is why they are called as the neutrophils are the martyrs of the immune system. Martyrs means they will first reach the site and they will attack the foreign material. They will undergo dying or apoptosis, and in turn they will attract other cells, other capable cells to the site so that the invader can be killed. So as we have discussed, phagocytosis has some discrete stages: activation stage, then chemotaxis, adherence, ingestion, and finally the destruction. Now, next type of immunity is the adaptive immunity. Adaptive immunity, it is also called as specific immunity. It is capable of recognizing selectively and it can eliminate specific foreign microorganisms as well as molecules. So it is different from the innate immune response. Adaptive immunity, they display four specific characteristics. First one is the antigenic specificity. Means they are adaptive immunity is specific to a particular antigen. Then second characteristic is the diversity. Third one is the memory. Immunologic memory means adaptive immunity, once it is mounted, it can memorize the invader. And fourth one is the self or non-self recognition. These four characteristics are very important, and if there is any breakdown of any of these characteristics, then there may be some pathological issues will come up, some immunological disease will be there if any of these highly regulated mechanisms fails in our body. Now, characteristics already we have discussed. First one is the specificity. The adaptive immunity is highly specific. Then diversity, it can respond to diverse antigenic specificities. Different types of antigenic specificities exist in the environment. So adaptive immune response can be generated against a diverse type of antigenic specificities. Then memory, this is very important characteristics. Adaptive immune response can memorize the antigen, so that if the same antigen attacks or comes for the second time, then it can immediately mount a vigorous response to eliminate the pathogen from the body immediately. And this response, memory response, this immunologic memory is also called as anamnestic response. This is also otherwise called as memory response or anamnestic response. Then self non-self recognition. This is a very important characteristics of adaptive immunity, because immunity is not mounted against the self body components. This is very important, and both immune system can recognize which component is own component and which is a foreign component. So that recognition is highly essential to mount a and immune response. So this is the overview how adaptive immune response works. Now, antigen presentation. Antigen presentation means in adaptive immunity, once antigens are recognized, it has to be presented through through some different molecules which are present on the cells. So these molecules are called as measured histocompatibility complex, or MHC. We'll be discussing in our subsequent discussions. But two types of MHCs are there. MHC1, it is responsible for presenting the intracellular antigen through the CD8 T cells, and MHC2, it is for the extracellular antigens through CD4 T cells. So this antigen presentation, it is mainly through the dendritic cells, which are major subpopulation or major population of cells responsible for antigen presentation. Now let us know the differences. As I said, there are certain characteristics which differs from innate immunity to adaptive immunity. So first one is the antigen exposure. So in case of innate immunity, antigen exposure is not required. It is not dependent on antigen exposure. But for adaptive immunity, it is essential. Antigen exposure is required. Then cells, different types of cells take part in innate immunity. That is the macrophages, dendritic cells, neutrophils, NK cells. Different cells, they take part in the innate immune response. But in adaptive immune response, mainly B cells and T cells, they take part actively to generate an immune response. Then cell receptors, they are preformed and directed against common molecules. In case of innate immune response, they exist as a preformed, but in case of adaptive immune response, they are generated in response to the foreign molecules which are coming to the body. These are generated in response to the foreign molecules specifically. Then coming to the onset in innate immune response, this is rapid. Onset is rapid. As I said, this is within hours or within very short time it can be generated. But adaptive immune response, it is onset is late. It takes around days to weeks. Memory, innate immune response has no memory. But adaptive immune response, it is memory response is present in case of adaptive immune response. Specificity, it is specific to common microbial structures, but adaptive immune response, it is specific to unique antigen. It is directed specifically against the antigen which is detected by the sentinel cells, and it has the response is mounted against that particular specific antigen only. Now, coming to the arms of adaptive immunity. Adaptive immunity, as we have already discussed, they are highly specific, and there are two different arms of adaptive immunity. One arm is called as humoral immunity, other arm is called as cell-mediated immunity. So humoral immunity, it is mostly the key cells are the B lymphocytes or B cells, and here B cells, they will first get activated, then there will be clonal expansion, different subpopulations of B cells will be generated, and they will be differentiated into plasma cells as well as the memory B cells. These plasma cells, they will be responsible for secretion of the antibody molecules against that specific antigen, and the memory cells, they will be responsible for the memory response of adaptive immunity. So this is how it works, the humoral arm. Then second arm is the cell mediated immunity. So here key cells are the T lymphocytes, and mostly this cell mediated immunity is mounted against the intracellular pathogens like viruses, and some bacteria are also there which remains as intracellular. Then cancer, transplant cells. So this type of immunity, this arm of immunity is active against this intracellular type of pathogens. So here Two subpopulations of T cells take part. One is helper T cells or CD4 T cells, then cytotoxic T cell or CD8 T cells. So the major part or major function of these cells, they will be recognizing the antigen in association with the MHC molecules or major histocompatibility complex molecules. Helper T cells, they will be requiring the MHC class 2 molecule, and cytotoxic T cell, they will be requiring the MHC class 1 molecule, and they will be in turn, they will be releasing different types of cytokines, and there will be cytotoxic killing of the pathogens by cytotoxic T cells. So this is how this cytotoxic T cell works. Then another type of cell, T cell, we have already discussed about the T regulatory cells. It suppresses the immune response, and it is responsible for maintaining the self tolerance, and it is also responsible for prevention of autoimmunity. Already we have discussed earlier. So this is the entire process in a flow chart, how adaptive immune response works. First, exposure of the foreign invader, then antigen processing and presentation will be required. Then it will be activated depending on the type of invader. It will be the humoral arm or the CMI arm, cell mediated arm will be activated, and there will be production of antibodies, cytokines, etc. And finally, it will be the pathogen will be, there will be destruction of the pathogen. Now, the based on the function, we can divide the entire immune response into different phases. So three different specific phases are there. This is for adaptive immune response. So that is specific, and it can be divided into three different specific phases: recognition phase, activation phase, and the effector phase. So recognition phase, here the binding of the foreign antigens to the specific receptors on the lymphocytes. Then this is the recognition phase. Then next phase is the activation phase. So once this binding of the foreign antigens to the receptors, it is complete, then activation phase starts. So here it will have a sequence of events which will lead to proliferation as well as differentiation of different cell populations. Then the final phase is the effector phase. So this is the final phase where the invader will be phase-wise eliminated through a specific programmatic manner. So either it will be through antibody production, or it will be through the CMI, or it will be through the complement activation. So finally it will be directed, effector phase will be directed for the final elimination of the pathogen from the body. So these three phases are highly regulated phases of the immune response: activation phase, recognition phase, activation phase, and the effector phase. So this is what actually I wanted to discuss today about the immunity and its types. So I think if you find these informations are helpful, please subscribe to my channel and give your comments. With this, thank you very much.